Hi everyone, welcome to another EAStalk webinar. Lallemand Animal Nutrition is our corporate member, and as a part of their membership benefits, they will present an exciting presentation today. Lallemand Animal Nutrition develops, produce and market high-value yeast and bacteria products and services solutions for optimizing animal performance and well-being. I would like to welcome Dr. Eric Leclercq. He is our presenter today. Dr. Leclercq earned his doctoral degree from the Institute of Aquaculture in Stirling in Scotland and has participated in various collaborative projects with the salmon industry. Eric is currently Global RT and Technical Manager Aquaculture at the Lallemand Animal Nutrition, where he conducts trials and provides aquaculture technical services globally. Eric is based at the moment in Toulouse in France. Today he will present a presentation understanding the potential of diverse yeast-based ingredients as preventive health tools for fish. My name is Anna Viskovic, and today I will moderate this webinar. The webinar will last for approximately 45 minutes with 30 minutes of presentation time and 15 minutes for Q&A. Please, during the presentation, send me all the questions you have for Eric. You can send it in a private message as well, and I will put it in the queue for the Q&A section after the presentation. Dear Dr. Leclercq, many thanks for accepting our invitation and for joining us today for this webinar. Please feel free to start presenting it.
Thanks a lot, Anna, for your introduction. Thanks a lot, everybody, for joining today. I hope you're having a good day, and we have quite a dense program, so I will stick to the point. We'll be happy to answer your questions later on, and I hope you will enjoy. So today, after some generalities, I will present the different type of yeast product from the wholesale yeast, can be live or inactivated, and the different yeast fractions, as well as their potential association. I will then move on to a few selected trials recent results before a little take home. The objective today is for you to better understand the different yeast components, their roles, and their contribution to fish health. And as well as to be able to differentiate the different type of yeast products, as there is quite a few around there, and it may be a little bit confusing. So first, a little bit of a history. The budding yeast, Saccharomyces cerevisiae, is amongst the oldest domesticated organism, which is traced back in Far East Asia, about 9,000 years before Christ. The first commercial use is believed to be circa 1700s for brewing, and the role in alcoholic fermentation was only discovered around 1850 by French Louis Pasteur. The first breeding experiment with yeast was done at the Carlsberg laboratory. Again, beer is around, and it was the first eukaryotic organism to be fully sequenced before the turn of the 21st century. More recently, we discovered that these little mammals heavily consumes spontaneously fermented nectar, which is good news. Since it is a living model of ancestral mammals, it may give clue to the genetic components and to the intrinsic benefit of alcohol consumptions and maybe yeast consumptions. As well, a different type of, in terms of yeast history, Saccharomyces boulardii was isolated from the skin of a tropical fruit in Indonesia in the 1920s, and it was found to reduce cholera, and it gave rise to, it is today a common probiotic with a range of medical application. So that's a little bit of background in terms of the yeast. Saccharomyces cerevisiae, in terms of commercial application today, this little organism has exceptional fermentative capacities and nutritional and functional properties. It is therefore extensively used across the food industry to ferment, first and foremost. Ferments a lot of beers, a lot of wine, and to bread 600,000 tons of bread, not bakery yeast, sorry, considering bread is quite light, that's a lot of bread. And as well to produce a lot of bioethanol from corn, for example, which you find back as a fuel for your car. As you can imagine, to do all these kind of applications, there is different yeast strains and different yeast species as well. But yeast is not only used to brew, it is as well used in terms of commercial application for a range of other things, for the plant farming of plants and cereals, as a biofertilizer and biocontrol. We can use yeast and yeast compound in the food industry in terms of savor ingredient, texture ingredient, cheese production, coffee and cacao fermentation, and as well there is a whole range of application of yeast for pharma and human health, including cosmetic. And another application that is of interest today, of course, is their use in animal nutrition as a feed material and as a feed additive. Animals have been fed yeast stuff for over 100 years. There is a large diversity of feed compounds as functional ingredient to support animal health and performance, and as well to provide some specific nutritional compound to the diet of animals. So yeast, what it is, it's a single cell organism, an eukaryotic, that is part of the fungi kingdom. It's quite small in size, 5 by 10 micrometer, which is around about the size of a red blood cell. And quite bigger than a bacteria, which is about 1 to 2 micrometer. It is very abundant and ubiquitous in the environment. Not all yeast are equal. There is a lot of different species, over 500 species of yeast, of which Saccharomyces cerevisiae is only one. And within each species, as you can imagine, of course, there is a lot of strains within each species. All these different yeasts have different morphology, physiology, metabolism, etc., which means different yeast products will have different properties. Looking at the yeast, it is composed of two main fractions, the cell wall, which is insoluble, and it contains, we'll come back to that, monooligosaccharides, beta-glucans, and chitins. And there is the intracellular component of the yeast, within the cell content, which is rich in protein, peptide, amino acids, glutamate, and nucleic acid. In particular, the yeast is quite rich in nucleic acid as a cell, and other little compounds such as enzymes. In terms of when we produce yeast, there is two main origins out of your fermenter. The fermentation can be run to produce yeast. So yeast is in this case a primary product, or the fermentation can be run to produce beer or bioethanol or other things than yeast. And yeast in this case is a secondary product. When you produce yeast, you therefore have the fermentation is specifically produced to produce yeast, to produce a specific strain of yeast, and under a specific process to target specification as regard with the yeast. So the quality of the yeast, the product is more consistent, it's qualitative, it's premium, and more secured in terms of what you get in terms of yeast. As a secondary byproduct of secondary product, it is a co-product or a byproduct from other industries. So your fermentation is run to produce a beer, not quite to produce the yeast. So you can expect indeed more variability in terms of the yeast composition, the yeast efficiency, quality, properties, specification of the yeast is much more variable since it's not your target. These products are regarded as more cost competitive. So when we produce yeast, and to go into the different type of yeast, you can start with a live yeast out of your fermentation. You get a yeast cream from which you can package, let's say, your live yeast, or you can go into more processing, a simple yeast inactivation process to get the whole yeast that is inactivated dead. or you can add a step of a further step of processing, the lysis, before the heat inactivation, which can be a simple autolysis or a more complete hydrolysis. I will come back to that in a few words, to get an autolyzed yeast or an hydrolyzed yeast, which is again a whole yeast cell inactivated but more processed. Another type is an enriched yeast, when you include in your fermentation batch different type of compounds that will be incorporated into the yeast before its inactivation. If we go further down the line of autolyzed and hydrolyzed yeast, then we can, of course, separate by centrifugation the yeast cell wall from the yeast extract. Lallemand, as a company, is a primary manufacturer of yeast with over 20 yeast plants across the world, mostly in North America and Europe. This is the core of the activity of Lallemand is to produce this different type of yeast, and the core of expertise, let's say, primary expertise of the company. So we will start with a couple of slides on the inactivated whole yeast cell, to show you the difference between the simply inactivated yeast compared to the more processed autolyzed and hydrolyzed yeast. So the inactivated yeast is an intact whole cell that is dead, but it's not processed. Autolyzed yeast, the lysis is done by the endogenous enzyme of the yeast. The lysis is triggered and induced by the endogenous enzyme of the yeast. So the yeast cell wall are slightly cracked, and the content is a little bit broken down. And on top of this endogenous lysis, we can add exogenous enzymes to further control, further process, further break down the yeast cell wall and its content to get a product that is more hydrolyzed. So along this increasing level of processing, from minor to medium to a deeper and targeted processing with specific enzymes that we can add for a specific duration, more controlled process. Of course, we will have a product that is increasingly more consistent in terms of specification, and also the crude content, the crude protein level, crude proximate analysis will show a similar, possibly similar parameters. You can of course see clearly that the cell being more broken down and the content more broken down, so digestibility and the functionality of the different yeast constituents will be increased, which is symbolized by the little schematic here, where we can see the yeast cell wall being more and more broken down. I come back to the inactivated yeast, where we can see when it is a byproduct. I put different colors here to really show that you may have different strains, different species of yeast. Maybe if you do a lager or dark beer, you may have different strains, so you add a layer of variability here, of course. So this whole different whole yeast have basically the same internal intrinsic constituents, but they are more and more available, more and more broken down and readily available by the animals. So the hydrolyzed yeast will be really your best product, the premium product to deliver the yeast. The yeast constituents, which will be small peptides, free amino acids, glutamate, nucleotides, and specific nutrients, which will be more available as well. So these products can be used as palatability enhancers, as the hydrolysis release some specific flavors, and there is a high level of free amino acids, which improve palatability and flavor. The free amino acids will as well help to maintain the gut in mucosal integrity. And the constituent of the yeast will as well serve as nutrients to support the gut microbiota. And as well, the nutrients and the nucleotide will have a role in immunity. The protein being, when the yeast is hydrolyzed, the protein being bioavailable and bioavailable and accessible physically and bioavailable to the animal, it becomes an interesting protein source. And this is here an example of a recent, the output from a recent collaboration where Lalaman was involved, showing where yeast was produced from a Norwegian spruce trees biorefinery to give a high quality feed ingredients that can replace more traditional proteins. So that was the first time yeast was produced on a larger scale, from a local sustainable resources as an alternative protein source. The project was conducted by the University of Life Science and NBU. And as I was mentioning, the yeast can be enriched with specific compounds during the fermentation before being hydrolyzed and autolyzed. And in this case, you have what we call enriched yeast. And I give, for example, the selenized yeast, whereby the inorganic selenium is added during the fermentation. And as the yeast multiply and grow its tissues and multiply, the inorganic selenium is incorporated into the cell by microbial immobilization, which is a totally natural process, of course. The selenium replaces the sulfur when the amino acids are immobilized in the yeast. And so from an inorganic selenium source into the fermenters, we achieve a yeast that is selenized and contains organic selenoamino acids, such as selenomethionine and selenocysteine in particular. And so the benefit of this type of selenized yeast is to get a natural source of organic selenium, which shows a balanced profile of selenium and a clean label as it is produced naturally by microbial immobilization. The role of selenium, of course, as we know, is quite wide and important in terms of antioxidant, with immune function, involved in thyroid activity, etc., etc. Reproduction and fertility of broodstock as well. It is an important part. And if you want to biofortify your end product, for example, it could be solutions. There is all type of enriched yeast, which can be enriched with different minerals, vitamins, complex, etc. Moving on to the yeast fractions, if we separate the cell wall from the yeast extract, starting with the yeast extract, the intracellular part of the yeast. This part is rich in protein, so the cell wall are not there anymore. So you can expect, of course, you just have the cytoplasm and all its constituents. It is rich in protein and valuable nutrients, small peptides, amino acids, vitamins, flavor enhancers, I was saying on the hydrolyzed yeast, and nucleic acid and nucleotides. These are two important components that are quite useful in the world of yeast nutrition. Nucleotide nutrition, it's an important application. Nucleotides are essential nutrients under some specific conditions, particular conditions such as rapid growth, tissue injury, recovery of tissues. Under those conditions, nucleotides can become essentials. Dietary nucleotides, therefore, can become a very valuable addition to the diet. They act as readily available building block for such critical tissues such as gut mucosa, white blood cells, multiplication, etc., where the body cannot synthesize de novo enough of them under these conditions. So the application of nucleotide nutrition is prevention and recovery, but clearly more targeted to recovery of the intestinal epithelium, as well, not recovery here, but more intervention of the immune response as an immune response enabler, as it will enable to multiply, to demultiply the red blood cell more efficiently, and as well as palatability feed intake promoter with this glutamate component. And there is, I will not go into the detail, but there is different type of nucleotides. You can have a source of nucleotide from hydrolyzed yeast, which will contain nucleotide, but other components from the yeast extract, from the yeast cytoplasm, as compared to purified source of nucleotides, which by definition they will contain only the nucleotides, or a less processed yeast biomass or yeast extract, which will show a different profile of constituents, of components. Moving on from the soluble cytoplasm of the yeast, now looking at the yeast cell wall specifically. The yeast cell wall is 30 to 50% of the yeast cell. It contains an external layer rich in monoligosaccharides, an internal layer of beta-glucans, which will be 20 to 35% of the yeast cell wall, depending on the strains, the fermentation process, etc. The moss, the glucans, and the little bit of chitins that you can find as well are microbe-associated molecular patterns, which are molecular signature which are conserved and present in microbes but not in the host. So this microbial pattern present at the microbe surface are recognized by specific pattern recognition receptor on the host. And so you can see here the link with the immune system. The main type of yeast cell wall product are the monoligosaccharides, basically, which are wall yeast cell wall, where you have the mannan and underlying on the mannan on top of the glucan layer. Typically, you find 20% mannan, sometime more in a good product. Then you can have a step at the factory in the yeast producers. You can remove the moss layer. In this case, you have exposed beta-glucans. The moss layer is removed. So, of course, you have virtually no moss, less moss, and more glucan, which can reach typically more than 30%. And once this moss layer is removed, can be a step of extracting and solubilizing the glucans. And so here you have a purified yeast beta-glucan product. And the content in glucan is even higher, reaching 60%. The main application for the mannans is gut and skin barrier functions, and for the exposed and the purified glucans, it's more about disease control, punctual intervention, which I will go back to it. Lallemand produces all these three types of products. So mannan microbe-associated molecular patterns, which is what we're in the business of producing for functional animal health. Of course, these little micro patterns are specific, diverse, delicate patterns, and so they need specialist tender loving cares or specialist processes and expertise to be produced in a functional and consistent way. And that means the process, of course, needs to be the fermentation and production process need to be very specific and specified and consistent, and be conducted with a selected yeast strain to achieve the targeted specification and functionality of the yeast. Fermentation-wise and post-fermentation, in terms of cracking the yeast, separating the yeast, spray drying the yeast or not, different type of processes to preserve the functionalities. And a big important step of quality and consistency to secure the consistency of the product. Moss, their main function is a bacteria blocker, and what's important to know is that the moss, the microbial pattern on the yeast of moss will ligate to the mannose receptor on the host, and this is not a danger signal, but a signal of immune stability, immune homeostasis, which will promote mucosal stability and good mucosal immunity. The application of moss are for gut and skin barrier function in terms of gut cytoarchitecture, skin response and skin mucus modulation, to increase the resilience and the maturity of this mucosal layer, and in turn the growth and performance of the animals. Their application is more for prevention, typically for prevention, and this type of product you probably want to supplement the animal continuously. As compared to beta-glucan, for which the function is strictly immune stimulation. Beta-glucan will engage with dectin-1 of the immune system of the host, and that will activate innate immune a range of innate immune components and facilitate specific immunity. The beta-glucans are recognized as non-self. It is a danger signal, and it is quite a strong pro-inflammatory component from which the macrophage will activate and multiply. The targeted application is clearly for disease control and vaccination response to secure, to promote a quick and stronger response if and when an infection occurs. You want to apply this over targeted risk periods, as there may be a risk of immune fatigue if you feed beta-glucans for too long. Of course, the yeast stimulation can be dangerous. And then this association of yeast fractions, where you can, of course, select different yeast fractions from different yeast strains to achieve your target application. In the case of Lallemand, we have this product that combines two strains of Saccharomyces cerevisiae with a non-Saccharomyces yeast. The different fractions, the different strains are separately produced and processed, and then they are combined. And these different fractions have been selected based on their morphofunctional features and screening, and the production process has been optimized for each of one of these fractions with the aim, the blueprint at the beginning to really go beyond simple mallan content and glucan content, but really go and optimize and maximize the power of these microbial patterns and associations. So by bringing together different strains and different strains of yeast and different parts of yeast, we bring in this product, for example, typically we bring a contrasted microbial pattern from different strains, so that co-activates a broader panel of immune receptors, including pro and anti-inflammatory pathways. So you synergize and you amplify the effect on the animals. The output, the result is the immune stimulation, the immune response you achieve is more coordinated as you crosstalk with you act on different immune pathways, and you work with different immune receptors and modulate different immune pathways which crosstalk and can synergize the effect of the product. There is no risk of immune fatigue, as this is not a dectin-1 beta-glucan immune stimulation per se. It is more complete and more balanced. So really this type of product, new generation type of yeast cell wall product, are really, you can be viewed as a next step in harnessing the power of the yeast microbial patterns. Yeah. And there is as well in this product the pathogen binding functionality that you find with the moss. I will give some few results on moss and the association of different yeast fractions and glucans on different species, quite a bit of salmon, zebrafish, and a bit of Mediterranean species at the end. So a trial here on seawater Atlantic salmon that is published, looking at the effect of specific moss product from Lallemand on skin health and sea lice protection. So the background is that we know that moss are very well described for their functionality and their benefit on intestinal health, but not quite so for their benefit on the skin. So that's what you wanted to reinforce, to check, to validate, and applying a sea lice challenge. So the salmons were seawater salmon of 250 grams. We applied the sea lice challenge and looking at two diets, a control and a diet supplemented with a moss product at 4 kiloton, which is a specific primary ground yeast from the company, of course, as you can imagine. We saw an improved growth, non-significant, of 11%, and a better intestinal C2 architecture, which is nice, which is what we know, what is very well recognized. So that's, of course, a good start in terms of improved intestinal health. And what we were more interested is to look at the skin barrier. So here you can see at the bottom the timeline. And the sea lice challenge was applied in between T1 and T2. Then we sampled 1 week after sea lice challenge and again 3 weeks after sea lice challenge. We find that feeding the moss product increases the density of goblet cell ahead of the sea lice challenge, which was 5 weeks of feeding the test diets. The skin mucus quantity was as well increased before the lice challenge, depressed following the lice challenge, and then at the end point you could see again an effect of the diet. So here we can see that with the diet we prepared, let's say, a better mucus secretion before the lice challenge. And then looking at the lysozyme activity of the skin mucus, we found that in the fish fed the supplemented diet, the lysozyme activity in the mucus increased more rapidly following the sea lice challenge, as it was significantly different from the control at T2, while at T3 lysozyme increased as well in the control and reached similar levels than in the supplemented diet. In terms of sea lice count, quite an important parameter as well. The level was statistically reduced at T2, showing a 17% reduction in the settlement of calamus. This is, of course, a single wave challenge, and we expect this 17% to amplify in real life conditions. So this study was interesting, showing that the benefit of moss goes beyond the intestine to the skin. The skin barrier was reinforced prior to the lice challenge and as well in response to the lice, for example, with the lysozyme activity that increased quicker following sea lice challenge. And that resulted in a protection against sea lice. Therefore, there is a potential to reduce the frequency of delousing and the impact of delousing if the skin mucosa is more robust, and we expect benefit in the field. Another trial looking at the same type of product, moss product on seawater Atlantic salmon exposed to a skin winter ulcer challenge with Mortella viscosa, a realistic challenge. In that it was not a LD50, it was a chronic level of mortality, low mortalities. I will tell you more. Seawater salmon fed either the yeast beta-glucan product at a commercial dose of 1.5 kiloton or a yeast moss product, again at 4 kiloton. So nutritional preparation was 40 days. Then we applied the BAS challenge of Mortella viscosa. Mortality was low, so we wanted to make the challenge to add an abiotic stress to amplify the bacterial challenge by reducing the temperatures and by increasing the salinity, factors that we know will add stress and increase the severity, the pathogenicity of the pathogens. And at day 53 here, we applied a 1-minute crowding with ideas to really destabilize the fish that were most compromised, followed by increasing of temperature to start a potential recovery of the fish. So that's a mortality curve. You can see that in the control, the mortality endpoint was 95%, survival, so 5% mortality, quite low. Nonetheless, it was significantly lower with the moss product. You can see in red here with an endpoint mortality of 99%. So in this group of supplemented fish were more resilient, in particular following the crowding here, as we can see that in the control that were really, there was quite an impact of this acute stress, crowding stress on the mortality, but not in the moss diet. And the glucan in orange here at intermediary levels. Looking at the wound score, as you can imagine, the winter ulcer challenge, we of course can perform scoring of the wounds. Looking at the mean score of the population, around throughout the trial post-challenge, the most fed fish had a significantly lower mean wound score. And if we look at more in details, at day 50, I pass on the first time point, but at day 50, we can see that in the mouse group, there was only 28% of fish that had an open wound, scored 2, 3, or 4, as compared to 41% in the control diet and in the glucan diet. So that was not significant, a trend of 0.12. And interestingly as well, within the wounded fish, so within this fish score 2, 3, or 4, we had a higher percentage of recovering wounds, so that is open wounds showing signs of cicatrization, that basically the wound was not active anymore. So here we had a clear significant effect on the fish that were wounded. Twice as many of these fish were recovering already. At the end point, similar pictures with a system significantly higher percentage of fish showing no wound or fully recovered wound, fully recovered with a silver layer, the wound was just fully cicatrized. So at the end point, a significantly higher proportion of fish, twice as much fish showing being healthy in the most supplemented group. And within the fish that were still wounded, again, we could see a near significantly higher percentage of fish recovering, with wound being recovering, not active. So that was quite a clear impact altogether of the supplementation with the moss and the glucan product showing intermediary benefits. Now we use the zebrafish model to look at mechanical inflicting a mechanical wound and looking at cicatrization of a strictly mechanical wound. There's no infection there. It was performed, of course, in a small-scale zebrafish setup, and so the medium was quite very low prevalence of infective agents, and so we really look at mechanical healing here. And we know that beta-glucans are known to promote skin wound healing in terrestrial animals when applied topically, typically for dogs and cows. It's quite documented, but not so much in terms of oral application of beta-glucan, and even less so for fish. So we fed zebrafish for 7 weeks, either glucan or this association of yeast fractions or non-supplemented control, applied a mechanical wound and look at recovery, first and foremost by their gross recovery that you can see here, which is a top view surface area of the wound, and we could see an improvement with the multi-strain yeast fractions, the association of different yeast fractions, the wound surface being significantly lower at the 16 post-wound. And now if we look at the granulation tissue, at the transversal cut of the wound, data were more, the impact was more visible. We do basically a transversal cut and measure the area of the granulation tissue. And you could see at day 4 post-wound. So you can see on the graph of the left, the visible impact on the top view was quite low, but the wound bed was basically filling up with granulation tissue, connectivity tissue, and recovering, closing itself. And with the supplemented diet with the multi-strain yeast fractions, the impact was significant, and the area of the granulation tissue was much higher than in the non-supplemented wounded fish, with beta-glucan showing intermediary levels. We'll have to pass on this trial. I'm afraid it's a bit late. Don't hesitate to ask a question on the potential of multi-strain yeast fraction and antioxidant product together that we show in this trial, that together they were able to basically reduce the level of chronic stress, just take my words on it. I'm not going to explain it, but reduce the level of chronic stress with the test diets and mitigate, give a better antioxidant status of the skin tissue. That was a very nice study that were presented at EAS this year. Reach out if you want to know more, looking at the impact of this combination of product across the stress axis of the animal. And in the Mediterranean species, we have a number of trials showing, in this case, the impact of Young, it's the commercial name of our multi-strain yeast fraction product, on gut and skin health. Basically, the product was fed for 6 weeks, and then we switched across group to look at the latency once we stop the product, if the effect did disappear, and if we could confirm the positive impact in the second group. And we looked at and we saw positive impact across the periods, across the different groups on the skin mucus quantity, a significant effect, some molecular markers at skin level of skin mucus productions, and that were in line with what we know a bit better about the positive impact of this type of product on intestinal health. So, to summarize my last slide, so Lallemand is a company that is specialized in the production, development and application of yeast and bacterial products. We are one leader in the field, continuously developing and providing premium quality products. Yeast-based products are natural, preventive health tool, in particular functional health tool. For aqua, we are not talking here about a silver medicinal bullet targeting a specific pathogen, but one tool to support the health of the host, which makes the pictures very different, but it's what of course, what is needed these days, and the benefit can be very, very strong. The importance of preventive health. Yeast cell wall are microbe-associated molecular patterns, and this type of product, of course, need a specific strain, specific production quality, high documentation, and the mining continue on our side to further expand and to further bring to you the power of yeast microbial pattern. Of course, to select which product you may need for which species, for your application, for your species, under your condition and your constraints. There is a shared expertise and a collaboration and discussion that is very beneficial to select the best product for your application, depending on a range of biological, logistic, strategy, and cost-benefit parameters. So with that, I would like to close with this quote, that we are not selling bread, we are selling yeast. So it can be a little bit agitating and disturbing sometimes. It's a live or dead microbe with a complex mix of molecules and content. I hope I bring a little bit of clarity on the different shades of functionalities and constituents of the yeast. I want to thank you all for listening. And here I am for any questions you may have. Don't hesitate to reach out by email whenever you want. You can find me via EAS or via LinkedIn. I'll be happy to help you. Thank you.
Many thanks.
Anna, here you are.
For the presentation today. You can hear me, right?
Sure.
Yes, it was a really interesting presentation. So you did a great job presenting your work and presenting the Lallemand Animal Nutrition. I'm opening now the Q&A section. So please send me all your questions for Dr. Leclercq, and I will make sure that I address it to him. So the first question we received is, has these products been used to improve for gills health in salmonids?
I'm not sure which product the person is referring to, but it's okay, I can answer anyway. This type of product, I suppose, is either mannan oligosaccharide whole yeast cell wall, or maybe the more advanced mix of fractions. We've been looking into it, but basically today the answer is no. To have a clear and short answer, we don't use this product specifically for gill health. We believe we have data on some effect of this commercial product here that already have some impact. When you look at molecular level in particular, we can see an impact on the gill health. We can suspect an impact on the gill health, but we probably need to bring it to go a little bit further with identifying and selecting maybe a new product that will be specific, that will target specifically the gills. As of today, it's not so clear with the products that are commercially available. Yeah.
Thank you, Eric. The next question we have for you is, how diverse is the number of species used in Lallemand studies?
Sorry, can you repeat?
Yes, just a second. How diverse is the number of species used in Lallemand studies?
I don't understand the first part.
How diverse, how many species you use in your research studies?
How many fish species?
Yes.
A lot. We work in general, like let's say in the last few years, you mean, quite a lot. You see, I've been presenting the zebrafish model. Atlantic salmon, as you can see, is quite a big one. Seawater and as well sometimes freshwater, of course. We've been recently doing a tilapia trial, trout trial with some of our partners that have been published recently, and we do once in a while work with trout. We've been working with carp lately. We've been working with pike perch. We've been working with tilapia. I'm not sure if I said so already. And of course, here I'm talking about fish, but we have a full branch of work with shrimp as well. So quite a few species, and this is only the aquatic ones.
There's a lot of species.
Yeah.
Yes.
With the different collaborators, we are under different setups and different geographies. Yeah.
Great. I will now read a comment we received from Somaya Shayadami. Sorry if I misspelled your name wrong. So the comment is, thank you, Dr. Eric, and thank you, Lallemand, for this pertinent webinar. Another question for you is, thank you, dear Dr. Eric. This product acts as mannan or oligosaccharides and or beta-glucans, I guess.
Yeah, I can see the questions. Let me go back to these slides quickly. Sorry. Up. This product, which I assume means this product here in green, the type mannan oligosaccharide, that's a point I tried to make very quickly. It contains both mannan oligosaccharides and beta-glucans. However, the beta-glucans are under the mannan oligosaccharides. They are not exposed. They are not exposed, hence they will not be. They will not go in direct contact with the host, because you have the mannan layer in between the glucans and the host. And so I believe we really need to think about this type of product as a mannan oligosaccharide product, within which the glucans may and likely they will contribute to the impact of the product, but you don't really have, you should not consider the glucan in this type of product where the glucans are underneath the mouth, as I said. They will not be as effective as a different product where the glucans are exposed or purified, because these ones, when the glucans are exposed, they will interact directly with the host. Here they are underneath, hence they will be less potent per point per unit of glucans. So although you may have 25% glucan, let's say, if your label here show 25% glucans, these 25% glucans will be not as effective as they will be on the next product where the glucans are exposed or purified. Sorry, long explanation, but basically for not much, but hopefully it's clear that this glucan in a monooligosaccharide product, the glucans are not exposed and less potent, but they may in part contribute to the product functionality.
Thank you, Eric. Another question we have for you is, what species are more susceptible, showing better response to Lalaman products?
That is a very broad question. None of them and all of them. I don't think we can put one species shows a better response to Lalaman product. And even to yeast product as well, I think it's quite clear, as I mentioned, Lalaman work with a number of species. The literature has been documenting the effect of yeast product across a range of aquatic and terrestrial animals. I think we are working here with quite a conserved mechanism, where the yeast cell wall, the yeast microbe will trigger some immune response in different animals. Of course, we know some species will respond maybe less, because maybe their transit is quicker as well. Not just maybe it can be because of transit time, not necessarily strict efficacy. And then we at Lallemand, of course, as you can expect, then we have to adjust the dose to achieve the level of biological response we target. So yeah. So I think that's the answer. Most species have the potential to respond to yeast, and Lallemand in particular yeast product. And the dose may need to be adapted, and in our recommendation, we of course take that into considerations. Yeah. Hello?
Oh, sorry. Yes, my microphone was muted. Sorry for this. So another question we have is, thanks for presentation regarding the sea lice attachment with moss. First trial presented. Why do you only report at T2? What about attachment at other time points?
Yes, T1 was before the lice challenge. So of course, there was no lice. Here we wanted to, the pre-challenge time point where we look where is the animal before challenge, and we can see more goblet cell, more mucus, and the same level, basal level of lysozyme activity. Then of course I report the T2, which is a calimus. Yes, the challenge was a copepodid challenge, so free swimming stage. This little free swimming copepods, they attach to the host, to the salmon skin, and they develop within a week in this temperature to calimus. So I show here the calimus level. And then 2 weeks later at T3, we were at the pre-adult non-sessile, the calimus detach is not attached to the animal with his filament. It becomes, sorry, I forgot the name, is non-sessile on the host, so it can potentially drop, and that's why I don't report the T3 level of pre-adult, because there was a drop. We could see that basically sea lice level had dropped across the control and the supplemented diet because of losses during handling. You would take your fish, you put it in a bucket, you do your sea lice count, and And in this process, you lose some sea lice that just detach and end up in the bucket. And so the data were not so clean. The trend was clearly there. It was just very similar, but it's open to critiques, let's say, and not reflective of the settlement per se of the sea lice on the fish and the diet effect. That's why I make long answers. Huh? That's right. That's a good question.
Thank you, Eric, for answering. We have anonymous comments. Thank you for explanation. You know what I'm willing to test in penne shrimp also. So I don't know who is the person behind this comment, but just so you know. The next one we have is from Marva. Have you tried to use selenium nanoparticles in conjunction with the yeast?
Ourselves? If you have tried to...
have you tried to use selenium nanoparticles in conjunction with the yeast?
No. That's a short answer. I'll try to make a short one. The answer is no, because we work with a selenized yeast. So when we work with selenium, we really focus on selenized yeast to understand and the specificity of selenized yeast. So no, I never worked with selenium nanoparticles.
Thank you, Eric. The next one we have is you studied the vast number of freshwater fish, but what about sea fish such as sea bass or sea bream?
Yeah, not sure I understand what is this question, in the sense that when we have a lot of data here on seawater salmon, which is a diadromous fish, but at the seawater phase, and I showed one slide here on the sea bream. So it is somewhere down, so I'm not going to go to it. So for sure, yeah, we have this slide I showed showing result on sea bream. It was a poster from EAS in Croatia or something like that. And we have other study on sea bass. And extra studies on sea bream as well. Sea bream and sea bass, we have quite a few studies here, looking at this multi-strain yeast fraction in particular, which combines different strains of yeast, bringing different immune patterns, interacting with different immune patterns. So we documented that quite widely on sea bass and sea bream in particular. In terms of intestinal and skin health. Yeah. And other fish as well, such as yellowtail. In Japan, we have a study and a few other ones. Yeah. So definitely, seawater fish are responsive to these products. Yeah. And shrimp. So this person mentioned shrimp as well. We have a few studies on shrimp as well, showing that shrimp as well are responsive, and cows as well. I mean, it's as I said, it's pretty conserved mechanism with specificities and dose, and some components may not respond in some animals, but the broad picture is quite conserved. Yeah. Sorry.
No worries, Eric. Thank you. The next question we have for you is from Valerie. She's asking, are Lalaman products thermostable and can be added to fish feed as a component before extrusion, or should we add them before coating?
Thank you, Valerie, for the question. It's a good question, very useful and important for you guys and for the fish farmer as well, to make sure he knows the product that works on the fish. In terms of yeast cell wall product and yeast product in general, inactivated yeast product, either for nucleotides, cytoplasmic content, or from the yeast cell wall products, I can say yes, it is definitely possible. Yeah. The extrusion, it is always applied extrusion when we do trial. and some of these products at least have been used many years, if not decades, pre-extrusions, and the biological benefits are there. So there's no. So the answer is yes. Of course, if we talk about live yeast or live bacteria or other type of product that Lallemand may sell, but not the yeast ones, here the answer may be no. But in terms of the product I showed today, yes, for sure it's okay, pre-extrusion.
Thank you, Eric. Barry's question was actually the last one for today. It's already 3 o'clock. So I would like to thank you all, especially to you, Eric, and to Lallemand Animal Nutrition for sharing all this interesting information with our members. I would like to thank you all for participating in today's webinar. Just a second, Eric, we still can see your slide. Okay. Good. I would like to thank you for participating in today's webinar. This webinar has been recorded, so probably at the beginning of the next week I will send you a link of the webinar for the video. And also you can download the Eric presentation on our website, but only under the member section. In a follow-up email, I will send you information about the next EAStalk webinar. Thank you so much, Eric, and thank you everybody else for being here. I actually received another comment from Marva, so just before we finish this, she said, thanks, Eric, the selenium in the salinized yeast might be of the size of nano, because I know from some papers that there is a green synthesis of nanoparticles using yeast.
Okay.
Yeah.
Okay, thank you. Yeah.
You're welcome.
Myra, don't hesitate to reach out if you want to discuss more about these things. Yeah.
Great.
I want to, yeah.
Yeah, tell me. Yeah.
So you cut me out. I really want to thank you on behalf of everybody, since not everybody can talk, and on my behalf in particular, and on Lallemand behalf. I think Anna and EAS is of great value to. Anna, you did a great job, and EAS is of great value and a very nice society for all of us. So I want to thank you.